Okay, so we're going to take a jab at another concept. The concept of let's do graded potential first. There's going to be a graph involved, unfortunately. Remember the picture of the postsynaptic neuron that I had here? One of the previous, yeah. So that postsynaptic neuron, just like any other cell in the human body, it has that resting membrane potential. Okay, this is RMP, resting membrane potential. And I have to kind of address, because I don't have enough space in that camera, doesn't allow for that. But in that, picture, we had neurotransmitter binding to the receptor, remember? So here's neurotransmitter binding to the receptor, and it opens the ion channels. Are you with me? Let's say neurotransmitter opens sodium channels, ligand-gated sodium channels. Where does sodium go? From outside to inside. So sodium, which is positively charged, flows into the cell. Everybody's on board? What happens to the charge, the membrane potential of the cell, in the cell? That's positively charged sodium gets in, diffuses in. Membrane potential of the cell becomes more positive. So this is your more positive. Okay? That makes sense? That's going to be a local change that is somewhat similar to the situation when you throw a stone in the water. So it creates disturbance, right? You create disturbance in the water. They have waves. But eventually these waves, they're going to disappear. Right? So same here. This local increase in membrane potential eventually will disappear. And go back to RMP. This increase is called, it's a graded potential, and it's depolarization. Okay? I'm going to put a title here. It's a graded potential. Okay? Now, by definition, depolarization is equivalent to excitation. I usually tell students to think of it this way. When cell gets more positive, it gets excited. That makes sense? What if the neurotransmitter that bound to receptor opens chloride channels? What does chloride go?
Chloride goes out to in.
Out to in. It's charged negatively, so cell becomes more negative. This is called hyperpolarization. And this is equivalent to inhibition.
What's the word after depolarization?
Excitation. So, cell becomes more negative, like receives negative news, and because it becomes inhibited. Does that make sense? Now you see the difference between neurotransmitter that's inhibitory and neurotransmitter that's excitatory, right? So excitatory neurotransmitter will open sodium channels or calcium channels, inhibitory neurotransmitter will open chloride, let's put it here, chloride or potassium channels, because when potassium gets out of the cell, it makes it more negative. Okay? Does that make sense? Now, one of the most important features of the graded potential, actually the term graded, means that this potential has a value. You open 2 channels, you have that much. You open 1 channel, you have that much of a change. Okay? It can have value. Does it make sense? Imagine. Remember the drawing of a neuron with all those dendrites? So each dendrite, say, forms a synapse, and at one synapse you have a graded potential, and then another synapse you have a graded potential, and third and fourth, and all those little tiny bumps, they kind of go up, right? And the question is, when does it become a signal? It becomes a signal when graded potential or multiple graded potentials together progress to what's called an action potential. This is really one of the concepts that's hard. I have an analogy, but I want to do without analogy first. In my experience, it works better when you do a proper physiological explanation first, then everybody says, what the hell is he talking about? And then we do the analogy, okay? So, I can get rid of this. Now, remember, here we're talking about ligand-gated channels. So this is neurotransmitter that opens, okay? Let's put it so that's sodium and calcium ligand-gated channels, or chloride or potassium ligand-gated channels. So now let's do, action potential. So this is your resting membrane potential here. And I'm going to kind of draw the initiation of this whole thing, okay? Okay, so you have a bunch of neurotransmitter that opens a lot of ligand-gated channels, and sodium rushes into the neuron. Are you following? Neuron becomes more positive. This is the change in the electrical potential around the membrane, right? And this electrical signal that is induced by a ligand, by a neurotransmitter, can be at certain points so high that it starts opening voltage-gated channels. Does that make sense? So you have that local depolarization here, and it becomes sufficient to open voltage-gated channels here. When they open, you have depolarization here. And it's so big that it's enough to depolarize the membrane here. Depolarization at this part depolarizes the membrane further and further and further, and you have the wave of massive depolarization running down the axon. That idea makes sense? Kind of. I see some kind of, I'm not sure about that, but we'll chat. Okay? So, at this so-called threshold level, voltage gated sodium channels open, and when they open, sodium diffuses in. I see that you know it, so I don't ask it. That makes sense? Now remember, they are voltage gated, so there is certain electrical signal that opens them. Turns out, here, the electrical signal closes them. Okay? Does that make sense? It closes them. Huh?
Is this the entire action potential?
Louder.
Is this the entire action potential?
Can you be even louder?
Sorry, is that the entire thing we're talking about now? The action potential?
This is action potential. Thank you for reminding me. I'm going to put like a title here.
Just wondering.
Oh, absolutely. You should do that. So, here on top, sodium channels close. Done. Close. And potassium channels will open. So now you have potassium doing what?
Going out.
Going out. When potassium goes out, how does it change the charge within?
Makes it negative.
Makes it more negative. So what you see is this. Now, while this step here is called depolarization, the return to normal, question? It's okay. The return to normal is called repolarization. Now look, this is your resting membrane potential level, right? You can kind of expect, probably we should stop here. We should, shouldn't we? Like, it's logical. Potassium channels are slowpokes. They don't close when the cell reaches resting membrane potential. They just stay open. They slow. They should close, and they will, but not right away. So what you have is a little bit of an overshoot. Does that make sense? So although cell is at the normal potential, potassium keeps getting out. And we have that little part here that's called, obviously, hyperpolarization. It's all going to make sense. And then your sodium-potassium pump, I told you we're going to mention it. It restores the concentrations, brings them back to normal. So let me go through the whole process one more time, slowly, and then I'm going to give you an analogy, which actually makes a lot of sense. So the depolarization, if the number of channels, when action potential is just formed, it's going to be ligand-gated channels. If the number of ligand-gated channels that are open sufficient to cause depolarization that will cross the threshold, you have voltage-gated channels opening. Does that make sense? Because at certain threshold they start just popping out. When this depolarization reaches its peak, which is, as you will standard. It's never like, it's never different. It's always 30 millivolts. Always. It reaches the peak, sodium channels close. Potassium channels open, and potassium diffuses out of the cell. It keeps on diffusing, 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 diffusing. Should stop diffusing at the resting membrane potential, but it doesn't. Okay? Because it's slow. The potassium channels are slow. So cell goes into hyperpolarization. Eventually they close right here, and sodium-potassium pump restores the ionic concentrations, and that particular part of the axon is ready for another wave. Got it? Now, imperfect analogy, but it kind of makes sense, that I use. And nobody said one statement. When I was talking about like depolarization here causes depolarization here, causes depolarization here, I was waiting for someone to say the domino effect. So imagine that you have dominoes that are aligned to produce the domino effect. But those dominoes are 500 pounds each. They're big, okay? So you come to one of those dominoes and you push it. And it's going to shake, right? It's going to shake a little bit. And then these vibrations are going to go away. Then you push this domino even harder. So there are vibrations, they're going to go away. And then you take a real good run and slam into it with your shoulder. And you flip it. And from that point on, the signal is going. This domino will flip another and go on and on and on. Does that make sense? Okay. So let's talk about some of the features of action potential. So first feature, really important, that I just said. It's called all or none. It can't be like there is no action potential that will be here or here. It's always the same value. Makes sense, right? Strength of a signal, I don't like to use the word strength, but I have to, is equivalent to frequency. Let me explain. So, actually, Miranda asked the question, like, what about like the amount of neurotransmitter, right? So, more neurotransmitter means more channels open. But the thing is, it doesn't mean that these dominoes will fall more. You can't fall. You either fall or not. All or none. You either have the action potential, but more neurotransmitter will produce more frequent action potentials. Does that make sense? So, if this is a medium signal, then this is a strong signal. Another example that I kind of try to illustrate action potential with, the idea of a sound. So I'm going to produce 3 sounds, and you're going to tell, and then we will compare them, okay? So I'm going to make a sound now. Can you hear it? I'm making a sound with my fingers behind my back. What does that mean? Am I making sound? Yes. Does it reach your ears? Yes. Oh, it does. Those vibrations, they do reach your ears. Do they reach your inner ear? Yes. Do they cause the release of neurotransmitter? Absolutely. But the amount is so tiny that there is no action potential, therefore you can't hear it. Okay? Now, can you hear this? That was kind of medium loud. Okay? So there was certain frequency of action potentials, right? That was louder. Make sense? Higher frequency of action potentials. Good? There's one thing that we didn't mention and nobody asked. I was very self-assured when with this action potential I was moving from left to right. And I was so assured that this wave is going to go from here to here. But why depolarization in this particular spot can't cause depolarization again here? Right? You see what I'm saying?
Yeah.
Why only channels to the right of me are being opened, but not to the left? What's going on? Well, it turns out, this is not I'm not going to write it, but some stuff we're going to put on the board. Turns out when sodium channels, when they close here, they aren't just closing, they're locked. Sometimes, when you park the car and you have to swipe the card. We had gated parking in a place where I worked in Louisiana. So you park the car, you swipe, the gate opens, you drive. If you will stop your car, the gate closes behind you. If you stop your car, walk out, and try to open it again, it's not going to work. Your card is kind of deactivated for 5 minutes. Does that make sense? So these channels here, they deactivated. Nothing can open.
Yeah, so it only comes out.
Say it again?
So it only comes out, it can't go in, right? Because it's locked.
Say it again?
So it can only, when it's at a certain point, it locks. So nothing else from the other way can come in. Is that-
So wave goes on my chair from left to right, right? So wave was here, and now it's here. Channels on this part are deactivated.
Oh, okay.
Nothing can open them. Nothing can allow sodium to go into the cell. Nothing can depolarize the membrane on that part. Does that make sense?
Yeah.
So this complete deactivation of sodium channels for a certain amount of time produces what's known as, it's right about that wide. It's called absolute refractory period. So basically, absolute refractory period means nothing can open those sodium channels. Okay? Now, let me ask you this. You need let's say X amount of neurotransmitter to get from resting membrane potential to the threshold. Okay? What about here? How much neurotransmitter you need to get from that bottom to threshold? More than X or less? More. See what I'm saying? So this part here is called relative refractory period. What does that mean? You can cause another action potential here, but you need stronger stimulus. You need more neurotransmitter to do that. So during absolute refractory period, tough luck. You can't cause another action potential over here. During the relative refractory period, you can cause another action potential, but you need stronger stimulus. Go ahead.
And this is to open up the, like the sodium channel
Yes,
again?
they capable now.
Okay.
So, like that example with the gated parking, if I would wait 5 minutes, I can open it. But since your cell is kind of down, you just need more to simply reach the threshold. Sodium channels are fine at this point. Okay, good? Okay, to make it digestible.